Okay. Hi to everyone who's listening on the recording. I forgot to start it until just now, so sorry about that. I'll post the slides on the Slack channel so you can catch up to where we are now. I think I made the slides text dense enough so that hopefully you'll be able to catch up. Okay, so here's the structure of the newly discovered antibiotic candidate by the authors of the paper in question. And they named it Halicin after HAL 9000 from 2001 Space Odyssey. Has anyone seen that film? I haven't. It's on the list. But anyway. So the initial screening data on which the model was trained was a 96 well plate assay of E. coli growth, grown at 37 degrees with shaking. The cells were grown for 16 hours with or without a dose of their screening library, which was mostly comprised of FDA-approved drugs and with some 800 or so natural products, which totaled about 2,300 data points once they'd removed all of the duplicates. And in terms of data analysis, they decided to classify hits or misses based on whether or not the growth inhibition reached a cutoff of 80%. So from their 2,300 compounds they screened, they found 120 hits, and that comprised their data set. Okay, here's a diagram that the authors include of how their neural network works. Now, I can appreciate that we haven't covered neural networks or machine learning of any kind in Python Club. So briefly, how to explain this quickly. A neural network is a type of machine learning algorithm, and machine learning algorithms in general are fairly general functions that you can fit to a large amount of data. So for example, in this case, the authors would train their neural network to make accurate predictions over the data set involved. The way you do this is the models will have a load of randomly initiated parameters, and over the course of training, they will see data, make initially a random prediction, and then they will update and adjust their parameters to minimize their error. And over the course of training, errors minimize and minimize and minimize until actually you have a fairly accurate model at the end. That's the basis of machine learning here. And the special features of the author's neural network, the message passing neural network, are that it works particularly well with chemical data, because, well, I suppose some background is that neural networks have sort of undergone a bit of a renaissance recently. That's what people call it, the artificial intelligence renaissance. That started probably in about 2010, 2014, something like this. I've lost my train of thought, sorry. Anyway, the special features of this neural network are that features for each atom are learnt based on their surroundings. So, for example, in the diagram here, atom 12, carbon 12, the feature for that atom will be a vector, which we can basically say is a list of numbers that will be some function of all of the surrounding atoms. The authors also included calculated features for each atom, like, for example, the hybridization for each one. Once a learnt feature is calculated for each atom, then they're pooled into a fixed sized vector that is a list of numbers that's some function of all of the atomic features that were previously calculated. And then that list of numbers is then fed into a neural network, which I will address another time. But the end result is it outputs a probability of a hit. After training, the ROCAUC score, which is the receiver operator curve area under curve, was 0.896, which is good. What's the ROCAUC score? In short, it takes into account the false positives versus false negatives. I think that's all I'm going to say about that for now. Okay, that's the only slide on the architecture of the neural network itself, but one day we'll come back to this. Okay, so with the trained neural network that was trained on the E. coli dataset that the authors generated in-house, they made predictions on the drug repurposing hub database, which I mentioned earlier is a database of drugs that were some way through testing for human therapeutics, be it animal trials, clinical trials, etc. This comprises a set of just over 6,000 molecules, and once you have a trained neural network, it's quite easy and quick to make predictions on new data. So on the new data, the drug repurposing hub database, predictions were made, and the 99 compounds with the best predicted activity were ordered in to the lab, and further testing was done. Essentially, they ran it through the original E. coli growth inhibition assay and found that 51 out of 99 of those compounds were actually hits, which is an all right hit rate. Then of those 51 hits, they used a separate neural network that was trained on the ClinTox database, which is essentially a large database which contains compounds and their clinical toxicity in various assays. And they used this to choose the compound with the lowest predicted toxicity. And the compound with the lowest predictive toxicity of the active compounds was halicin, this kinase inhibitor. Then further testing began, and boy, was there a lot of further testing. So the first question they wanted to ask was, is it bactericidal or bacteriostatic? So they did some time and concentration dependent killing assays. That's concentration of halicin in rich and minimal media. Now the idea here is if you can kill a bacteria in rich media, but not in minimal media where it wouldn't be replicating, then it's bacteriostatic, which means it kills by preventing replication, or maybe kills isn't the right word. Otherwise, if it kills in both conditions, then it's bactericidal. And what the authors concluded from this is that it is bactericidal. Then they gave it some resistance challenges. Part of the aim of these experiments was to determine if there's a known mechanism of action. If one of the resistance genes conferred resistance to the E. coli's, then that might give some clues as to the mechanism of action. Otherwise, it's a good experiment to see how viable this antibiotic would be in the long term. So they had a library of known E. coli resistance genes, and they were all transformed into E. coli individually, and each one was screened against various concentrations of halicin, and none of them had any effect on the minimum inhibitory concentration of halicin, which essentially means that no known mechanism of resistance was able to prevent halicin from working, which is pretty interesting. Resistance challenge 2. So resistance challenge 2, they actually had clinical isolates of some particularly troublesome bacteria, which they say, I think they had it gathered from a WHO list of the world's worst bacteria or something like that. The list probably has a different name in real life, but that was the gist. So, for example, we have suspects such as tuberculosis and several drug-resistant bacteria from other species like Pseudomonas aeruginosa. And they found that only the Pseudomonas aeruginosa showed some sort of resistance towards this, which they speculated could be due to a lack of membrane permeability of their drug, something which we'll get onto later. Okay, then they proceeded to some mechanism of action testing. One way that you can test for a mechanism of action of an antibiotic is to see if you can evolve a resistance gene for it, and then that might give you some clues as to how the antibiotic works. So on both solid media and in liquid media, the authors gave E. coli continuous exposure to the antibiotic for either 7 or 30 days, depending on whether it's on solid or liquid media respectively. And they found in both experiments there was no spontaneous suppression mutants, which on the negative side means that they were no closer to finding out how this thing works, but on the positive side, it sort of backs up their previous finding that resistance evolution to this bacteria is to this antibiotic is pretty rare. The next battery of tests was a transcriptome response in response to halicin. So that is, they measured the transcriptome of E. coli in a dose-dependent manner to halicin to see what correlates with an increasing dose of halicin, see how the cell responds. They found that cell motility genes were downregulated, so that's like flagella and stuff, and iron homeostasis genes were upregulated at sublethal concentrations. The implication, they say, was that the downregulation of cell motility genes could implicate a disruption in membrane potential, which is essential to maintain respiration. So a higher concentration on one side of the E. coli membrane of protons than the other will sort of encourage a flow of protons through ATP synthases, which is essential for respiration. And if you disrupt this balance, then it prevents the E. coli from being able to respire like normal. Then they followed this up with extra experiments, like, for example, changing the pH of the media to see if that has any effect on the efficacy of the antibiotic, and it did. As you increased pH, the potency of halicin decreased. And they followed this up with a fluorometric assay with this DISC-3 fluorophore, and you can follow the link on the reference there to find out more about the assay and how the fluorophore works. But I think the fluorophore got activated in response to healthy pH homeostasis when challenged with a pH change, and it doesn't respond if you've disrupted the healthy homeostasis of the cell membrane. And they found that the antibiotic did disrupt the healthy homeostasis of this membrane. And they also found that it synergizes with iron whilst they were testing whether or not it synergizes with other antibiotics. And the minimum inhibitory concentration of halicin was increased as they increased the concentration of iron, which gave them more clues as to the mechanism of action. They found that was similar to ionophore style antibiotics, which apparently binds to certain ions and then disrupt the transport of ions across the membrane. And here we go. Here's the likely mechanism of action. Halicin may bind iron prior to membrane association and proton transport disruption. What exact protein target it affects, not sure, but at least we've narrowed it down to a sort of sliver of mechanisms of action. I'm not a very good microbiologist, so I might have overlooked or misquoted some pretty key information, so please check the paper if you need any clearing up on anything. Okay, and then they did some animal testing with a sample size of about 6. Both of these tests that they do are pretty grisly. So they made a 2 centimeter squared wound. That's supposed to say squared, not cubed. 2 centimeter squared wound on the back of mice, and then inoculated it with, I think that the A was Acinetobacter or something like that. But it's a wound infection, multi-drug resistant tissue necrotic bacteria. It's sort of like a flesh-eating type thing. It's pretty horrible. Anyway, they inoculated these huge wounds on the mice with this bacteria and then treated them with or without the halicin. And they found that the carrying capacity of the wound, that is how many bacteria can actually live there, or how many of these infectious bacteria can actually live there, got reduced by a few orders of magnitude, which was pretty encouraging. The second test was a C. difficile test, which if you come across C. difficile, then you'll know that it commonly comes up after you've been on a course of antibiotics and your gut flora has been sort of abolished, which lets this C. difficile, this opportunistic pathogen, sort of take hold of your gut and can result in some quite nasty consequences. So in this case, they sterilized the guts of these mice by giving them a course of ampicillin over a few days, then a day to recover. And then they force-fed them with 5 times 10 to the 3 C. difficile spores with a gavage, which I found out what that was. It's sort of a force-feeding tube, which is horrible. And then they treated them with or without either halicin or metronidazole, which apparently is the first-line antibiotic for C. difficile treatment. And they found that it was actually, halicin actually outperformed metronidazole in this particular test. Of course, it's all subject to further testing by independent bodies and larger sample sizes and so on. But it's all quite promising. So the conclusion: the ChemProp model is a message passing neural network that allowed the discovery of this new antibiotic. It was trained on a relatively small in-house data set, and then the predictions, it could extrapolate quite far beyond what it had been trained on, and then be used to identify antibiotic candidates from existing databases. The message passing neural network, they found in this paper and in other papers, is quite effective because it can generalize quite far beyond the data it's seen compared to other similar types of models for similar types of data. The source code is freely available online. You can check that link to check it out. And the other conclusions were its mechanism of action. It's probably disrupts membrane proton transport. It's effective against pan-resistant bacteria, and it's fairly effective in mice. That's all of that presentation. Briefly, are there any questions?
May I ask a question, which maybe is not related to the machine learning?
Yeah.
Is when you go into screen, when you are talking about the neural system, neural something, the theoretic behind it, it's like I think it's slide 2 or 3, when you show there the 3 red lights and then the structure of one compound. Go lower, 8. I think slide 8.
Yeah.
Yeah, here it's like basically how could you decide which is the bad, or you mean it's basically the how you decide the selection conditions. It's like based on your knowledge. For example, you know that at that position, if you add 1 hydrogen or if you add 1 OH or whatever, it will not get any function or whatever. How could you select your compound?
Yeah, that's a good question. So one of the advantages of machine learning is that you don't actually need expert knowledge or domain knowledge about what you're doing. So for example, the neural network that was trained here saw nothing more than the compounds and the result of the assay, and it was completely naive to everything else. Could you ask me the question again, please?
Yeah, for example, like what I'm thinking, maybe, okay, from my understanding, and then if you want to identify if this if a new compound is really functional to this disease or not, it will depending on if you do some modification based on, for example, like the position 12. If you add 1 hydrogen or add 1 OH, yeah, OH or something. Yeah. And then how do you know if it will be work or not? I mean, based on what kind of knowledge you will know that?
So the neural network will only be able to make predictions, and the predictions will always be somewhat subjective. Estimations, let's call them. It will estimate the efficacy of a new compound, which is why the authors of the 99 compounds that they predicted might work, they tested all of them and then confirmed the activity of 50 or so of them. So I suppose that shows that it's not 100% accurate, but it's accurate enough to be useful. I think there's a saying that all models are wrong, but some are useful. And in this case, it is a little bit wrong, but it's still accurate enough to be useful. Does that come close to answering your question? I may have sidetracked a little bit.
And I think maybe, it's like I need to understand a little bit more about the machine learning. I mean,
it's about the parameters that you're training it on and how you tailor it to if you have change the functional groups and stuff, and how that would, what if you have to change the parameters that you're constantly training your neural network on. Do you know what I mean? Does that make sense? Is that
I can a little bit understand it. It's basically like you start some parameters or you start some conditions, and then you play around with these conditions, and then so according to that you get the score, and then you set a cut line. Say, okay, if it's higher than 0.88 or something, and then you think these ones are really function, and then you take these ones as the candidates.
Okay. So the thresholds the authors would have set themselves probably arbitrarily. I imagine they would have made all of the predictions before setting the thresholds and said, okay, 0.8 looks good. We can that's an amount that we can practically screen. So in terms of setting a threshold, the authors would have done it manually. In terms of the parameters of the network, the updating and adjustment of the parameters is completely automated, with some algorithms that I'll get into another time. Does that satisfy your question?
Yeah, I think so. Yes, so thank both of you. Thanks a lot.
Cool. Okay, so very briefly, I'm conscious of time going on. I'll share these slides with you. All of the links here are live, so it should take you straight to the papers. This link to the source code is also live. I don't think I put in links to the original papers here, but I can do that. I can do that in a minute. And if we follow the link to the source code, we get taken to this website called GitHub, which is a public code repository is pretty much an industry standard to publish your code on here. Definitely the University of Manchester has a lot of stuff on here. And this is the chemprop page. It comes with a README file, readme.md, that gets rendered here. And we can see here that the author, Kevin Yang, has made a new repo that contains COVID-19 data sets. So the data that he's looking at are actually SARS-CoV-1 data sets that he's sort of acquired, cleaned. He's got the assay ID here, which we can search in PubChem if we want to know how this assay works. And he's put together quite a lot of data from various sources. Also in this README file on the original chemprop page, we have options of or details on how to install this chemprop model on your own computer. So for example, how to install it with Conda. Docker is an environment managing tool as well, so that you can make sure that you have all of the necessary requirements and no clashes. He's also got a link to a web application, which is available here. It's not available here. Well, never mind. If you want to download the whole of this repository, then one simple way to do it is to click on this clone or download from the landing page, and you can click download zip. Or if you have Git on your command line, you can type something like git clone and clone the whole thing to your local repository, or to like a local folder. So in this notebook that I made here, that's exactly what we do. If you're wondering how to go switch it to dark mode, then under tools and settings, there's an option for light or dark mode. I'll leave it on light mode for you for the sake of making it a bit more readable. So I'll do a quick whiz through of what I've done here. I will continue to add to this over the next day or two, just so that there's, so it's a bit more usable. But essentially, the scope of this notebook is to download some of this data from various old SARS-CoV-1 assays. And we'll have a look into it. And then we do some brief analyses, and then briefly train a simple machine learning model on the data, and then that's pretty much as far as I got. The steps after that will be validating the machine learning model, investigating it a little bit, and then using it to make predictions on new data, which I'll be adding in the next couple of days. So it's a pretty messy notebook so far, but you can see initially I've put in the exclamation mark before this command, and we remember that the exclamation mark is a character that escapes Python and enters a command line command. In this case, the command line language is bash for a Linux machine. Git clone is the command to download straight from GitHub. The appropriate link you can find in this clone or download, and it will copy it here. It's different from the link that you'll see in the URL bar. Especially it's got this .git at the very end. Can I see it? No. Anyway, so that's what I do here. The other command is to quickly install a tool that makes a tree structure of the installed directories. Do I do that? No. Well, anyway. These cells cover something that we saw before. It quickly downloads Conda, which is the package manager that isn't installed by default on Google Colab. It makes it executable, it runs it, and then installs RDKit and py3dmol. That takes a couple of minutes and creates quite a lot of output, which you can use for debugging if you want, but you don't need to see it. So if we want, we can minimize that. And then in this cell, we append to the system path the file where we've just installed RDKit, just so that the Python interpreter knows where to look when we call either py3dmol or RDKit. And here we have a structure fetched from the PDB using the py3dmol widgets. It's the structure 6W9C, which is one of the papain-like proteases from SARS-CoV-1, which is the target of the screening data that we'll be looking into in this tutorial. This particular structure is a trimer, so I've highlighted one of the units with you know, a surface and some cartoon representation. In this cell, I read in the data, or the screening data that we've just downloaded using that git clone command. So that git clone command basically downloaded a couple of folders worth of data. So this is the path from where the notebook starts to the data, to plpro.csv. We print the length of it, and we can see it's 230,000 data points. And then we have a quick look at the head, and we see that the columns are called smiles and activity, which is either 1 or a 0, according to a hit or a miss in this particular assay. I'm going to include some more stuff on how the assay actually works at some point, because we have access to that sort of information. In this cell, we have a look at the percentage of hits out of the entire data set. So we take the activity column, we sum it, and then we divide it by the total number of data points, and then times it by 100. And we print that out here in an F string, which is the type of string where you can drop in variables, which is pretty handy. And we can see that the percentage of hits are very low, which is an issue for machine learning. For example, if you only have 1% hits and you train a model on that data, it could be 99% accurate by predicting miss every single time. So that's an issue that we'll have to overcome later on in this notebook. In these cells, we import RDKit, our favorite cheminformatics library, and we look at the scaffold of each of the molecules in this data set. A scaffold is basically the core of the molecule without any decorating groups. I'll find a more rigid, robust definition by the time I present this for real. But essentially, all the decorating groups, like for example, a methyl or a chlorine, will be stripped off, and it will return you the smiles of the core group here. And then we count the values of each one. This is the smiles code for benzene, and we can see that that's the most commonly occurring scaffold in the entire data set. And in this cell, I tried to draw them, and this exact code worked in my local notebook, which I can show you now. But it didn't work in the Google Colab notebook for reasons unknown to me. Here we go. So here's what I was trying to achieve. So it renders in a nice grid each of the molecular scaffolds, and the label underneath is how many times each one occurs in the entire data set. So we can see benzene occurs quite a lot of times. That's nice, popular. And we have a few more. And I only rendered the first 10 because I think there are a few thousand unique scaffolds, and I didn't have enough room to render all of them. Back to the notebook that I'll show you. So here's what didn't work, and I'm still struggling to understand why. So I'll sort that out next time. This is me attempting to solve that problem. Also didn't work. And in the next few cells, I essentially try and isolate out just the benzene-based molecules, the molecules with a benzene scaffold, by sort of searching the scaffolds by which ones are equal to benzene. And then I decided to build my model based on exactly that. I'll scroll through these ones fairly quickly. In this cell, I have a little look at all of the hits and see which scaffolds are most common amongst the hits. Do my best to render them here. The lack of a scaffold here, I think, pretty much corresponds to a molecule without a scaffold. That is, like, for example, nitrous oxide, I don't think has a scaffold. But again, I'll need to come back to you with a more formal, rigid, and robust definition of what on earth a scaffold is. Here I locate compounds containing benzene. I've done my best to annotate what I'm doing here, but again, in the interest of time, I'm just doing a whistle-stop tour through what I've done. If you want to steal any snippets of code, then please feel free. But yeah, just note that it's an unfinished notebook that I mean to finish off in a future tutorial. And here we start entering into modeling. So in this cell, I use a technique called upsampling to overcome the class imbalance. So we remember that the minority of compounds were actually hits. So I try and overcome that. And the two ways you can overcome that, well, essentially I want to end up with a data set that has the same number of hits as misses. So I can either randomly sample from all the misses, which are the overwhelming majority, randomly sample a number of them equal to the number of hits. So now I have an equal number of hits or misses. That's called undersampling. The alternative is oversampling, which is pretty much the other way around. I randomly sample and then resample hits until I have the same number of hits as misses. Now, in this case, naturally, you end up with a lot of duplicates of each one, which is fine-ish, but I'll come back to this more in the following tutorial. And then in these cells, I generate molecular fingerprints for each one. Molecular fingerprint, very briefly, is a vector of ones and zeros, a little bit like this. And each 1 or 0 corresponds to the presence or absence of a particular chemical group. And which chemical group each 1 and 0 corresponds to is a matter of how the algorithm was designed. Usually they're hand-engineered features, I think. But again, I'll come back to you with more info on that. And we can also visualize which part of the molecule each 1 and 0 corresponds to. So hopefully I'll be able to do that by the next tutorial. We make Smiles fingerprints as fast as possible for every single Smiles code in the dataset, and we do that by using something called the Pandas apply function, which is essentially, say I have a column, in this case Smiles, that I want to do an operation for every single cell in it. Rather than writing a whole for loop, I can do dot apply, and then to that dot apply function, I feed another function, in this case Smiles to fingerprints. Notice I'm not putting brackets like that on the end of it as you normally would with anything else. I'm just giving it an empty function, and essentially then Pandas will handle everything in terms of making sure that each Smiles fingerprint in that column will get this function applied to it. And you can do this with pretty much any function, with some limitations that I'll come to later. It's quite a time-consuming operation, so I also start a timer and then stop it after it's finished and print out the time here. And we can see it took about 45 seconds on this Google machine to do it. It takes more like 90 seconds on my computer here. We start splitting the data into a training set and a test set. The reason for that being is that we need a training set to do our parameter adjustments on our model, which will be imported later on. In this case, we'll be using an ensemble model called a random forest. But I'll come back to that in just a second. In this cell, we split our data into a training set and a test set, where the test set is roughly 20% of the data, or in this case, exactly 20% of the data, and the training set is the other 80. And the idea is the model will adjust its parameters to minimize its error on the training set. The process of adjusting its parameters to minimize error is exactly equivalent to training. You can use those two terms interchangeably. And then you validate the model on the testing set. And the idea there is that you can, one, make sure that you're not predicting a load of nonsense on new data, and you can prevent something known as overfitting. And overfitting is basically where you make a model, a machine learning model, with far too many parameters that analogously learns all the answers to what you've shown it, and then fails to generalize to anything beyond the training set. So that's why we have a test set to try and make sure that that doesn't happen. It's sometimes common to have like a third partition for a sort of a final validation step, but again, we'll come on to that another time. And in these cells, I import a random forest classifier. And a random forest classifier is, as the name might suggest, it's a classification algorithm. And the random forest part refers to the fact that it's a large group of decision trees. And a decision tree is an algorithm that is sort of tree-like. So for example, I tried to draw a little ASCII diagram of it here, but say we have an input. Let's say we feed this decision tree just the log p value and I don't know, the molecular mass of a particular compound. In one decision node, it might say, is the log p greater or less than this particular value, this particular parameter that will be learned over the course of training. And if it's greater than or less than, then it will get passed to one of the next decision nodes or the other, which also contain a learned parameter that corresponds to potentially the same the same metric, like log p in this case, or maybe to the other metric, like molecular weight, I think I said it was. And after a few steps, then it will return the predicted output, either a 1 or a 0. That's a oversimplification of decision trees for you. And essentially, a random forest is a large collection of said decision trees. That's all. They're all trained on the data, and the output of the random forest is a sort of a vote between all of the decision trees in the forest, which has a few advantages. Partly that each model, each individual tree might learn to identify slightly different features rather than focusing on one in particular. And also you can measure the variance in the prediction from the different models, which you can use as an uncertainty measure. So you can tell whether or not your model is confident or unconfident about its predictions, which is pretty essential if you're going to do anything like act on any of the decisions made. You need to know whether or not your model is certain. I'm using the scikit-learn library, which is pre-installed with Anaconda, so you shouldn't need to worry about downloading it. sklearn, it's got some pretty great documentation on the internet. sklearn, let's have a look at random forest. I recommend this as a go-to library if you're thinking of doing any machine learning style analyses. It's also got a lot of other functions that are useful, like, for example, principal component analysis and linear regression, many more. It's got a pretty extensive user guide and a lot of examples. And here on the documentation, we have information about the parameters that the model takes in. A lot of these are optional, But for example, we've got n estimators, number of estimators, which is the number of trees. The criterion, which I'll have to come back to you about, and a bunch of other parameters that are all optional. It tells you more about each of these parameters on this list here, and further down it will tell you once you've initialized the object what attributes it has. An attribute is an object that belongs to the random classifier, and we have more notes about that, and most usefully some example snippets of how to use it in action. So for example, we can see that in these lines of code, the authors are making a randomly generated classification data set for the sake of example. Then they are initializing a random forest classifier under the name CLF here. So after importing it here, they initialize it by essentially typing it in with open and closed brackets, and that will initialize it as an object. They also feed in some of the optional parameters, like for example, the maximum depth, which refers to how many layers of decision nodes you want. And the random state. Random state is like a seed to the random number generator involved. And normally you don't have to worry about that. But if you want to replicate exactly what these people have done, then that's a reason to include your random state. But for our purposes, I don't think we need to worry at all about what a random state is. And training the random forest classifier is as simple as clf.fit, and you give it x and y. In our case, we'll be calling fit on x train and y train, that training partitions of our data. X referring to the fingerprints that we generate, and y referring to the activity level for each corresponding chemical. Training is fairly quick, and you can also look at what's called the importance features of each input, which is cool, especially if you're dealing with chemicals, because if we're doing a QSAR, then we can look at the importance of a particular chemical group, which is something that's useful for a synthetic chemist, for example. They can see that such and such a carboxyl is good and such a chlorine is bad. And then you can call predict with this object now that it's trained, and feed it pretty much any compatible data. So in our case would be feeding our model that would have been trained on the SARS-CoV-1 PL Pro dataset. We would be feeding that some drug repurposing hub data to try and estimate which drugs from that dataset might be effective, at least in this assay. To predict factors beyond that, like for example, how safe they'd be in clinical testing, would need other models, but we can at least make a prediction about how accurate, or sorry, how effective something will be in this particular assay. It all depends on what data we feed to the thing. So here we can see we define this classifier under the name model. We initialize it, and we feed it this optional parameter, number of estimators 10. So that's 10 trees, and I don't give it any other parameters. So everything else will just be default. Here we train it, model.fit, and we feed it the training partition of xtrain, and that's the features that we generated, the fingerprints, and the corresponding active or inactive column from the data frame. And then to evaluate, we make a prediction on the partition of the data set that we save for model evaluation, X test in this case. We're calling that ypred. So, and then we stuff that into a data frame of which compares the predicted and actual for each of the for all of the test sets. So these are predictions that on compounds that the model's not seen before, and you can see in the first couple of rows it seems to be getting it right on the whole. And this is where I get a little bit suspicious. So I calculate an accuracy score based on the sum of rows where predicted is equal to actual, and then divide it by the total number of predictions and times it by 100. And at this point, I'm suspicious, and I was gonna ask Steve, what on earth am I doing wrong here? Because that's far too good to be true, I think. And I start running some diagnostics, and we're reaching the end of what I managed to do in the time that I had. So I plot something here called a confusion matrix, which essentially plots the true negatives against the true positives. So predicted hit, predicted misses, and actual hits and actual misses. And I put that into a color grid as well for some, some visual aesthetic, but essentially it's exactly the same information as we have here. And then I go into more depth plotting the receiver operator curve, which I mentioned in the presentation, which is how the authors of the message passing neural network evaluated their model. And again, I'm very suspicious about this. This is pretty much the absolute best you can get this curve. So the idea of this game, the receiver operator curve game, is to get as much area under the curve as possible. And I've made pretty much a right angle, which means I've maxed out on how much area I can actually get under this curve, which again leads me to suspicion that I've done something wrong. So tune in next time to find out exactly what I did wrong. And I've got some to-dos here as well for myself. Apply the predictions to the drug repurposing data set. Find out what's wrong, question mark. And then map the contributions of certain chemical groups to the chemical structure, so we can actually visualize which side groups of this benzene set that we're testing are actually effective. Well, anyway, that's all I have for now. Hopefully I'll have this ready either by next Friday, or because it's mostly done anyway and it aligns fairly closely with what I'm supposed to be doing, I might be able to squeeze a midweek tutorial in. I think I promised someone else next Friday to cover more BioPython, because they're very interested in sequence-related data. So maybe I'll cover that. I'll keep you updated on when I plan to do this, and I'll record the next one, and I'll actually start recording it from the beginning next time as well. And that's all I have for now. Does anyone have? That's all right. Thank you for coming.
I'll have a look through it and see if I can spot any errors. But it does look very strange. You get perfect results.
Yeah, I wonder if it's a result of the oversampling.
I guess it just oversampled like 2 incorrect things. Like if there are only like 2 things that were.
Yeah, I think maybe in this case undersampling would be a more safe tactic, in which case I think that will leave me with a data set size of about 20, 30, 40, which is small, especially considering I've got something like 2048 different features for each molecule, which is a little bit too many. Ideally, I want to have more rows than columns in a machine learning data set.
Well, you could do lots of it. You could do lots of undersampled tries and take the average of the result.
Yeah, this is true. So for example, train a model on each small chunk, and then use that as another ensemble model. Is that right, Steve?
I'm thinking.
Sure.
Okay, thanks very much, James. I think I'm going to have to leave now. So
yeah, of course. Well,
thank you.
Thank you all for coming. Thank you, Kath. I'll see you all soon.
Yep. Indeed.
Yeah, cheers. Thank you, James.
Thanks, James.
Thanks, Magnus. Thank you all.
Bye.
Yeah, I'll head off now as well.
Okay.
Come back to you with a hopefully working tutorial on maybe midweek. How's that sound?
Okay.
Sounds good.
That sounds great. Thanks.
Thank you.
All right. Bye, everybody.
Bye.
Have a good week.
Bye-bye.
Thanks a lot.
Bye.
Bye.